In last lecture, we were discussing the modes of transmission of hepatitis B virus. In that, I forgot to mention a very important point that it can also be hepatitis B virus can also be transmitted from mother to child while childbirth process. Today, we will discuss the immunopathogenesis of hepatitis B virus, right? And along with that, we will also discuss the mechanisms by which hepatitis B virus evades the immune system of our body, right? So let's suppose this is a hepatocyte that has been infected with virus, and as it is infected with this hepatitis B virus, it will generate lots of not only virus particles but non-infectious subviral particles which only consist of this envelope, right? And of course it will also create e-antigen that have not been shown in this diagram, right? So all the cells, right, all the cells of our body, they have to process and present the proteins that are being generated, that are being synthesized in them on their special receptors called MHC1 receptor, right? A normal cell will show a normal protein, right, normal body protein, and a virus infected cell will of course present viral protein, fragments of viral protein or MHC1. Remember, it does not present the whole protein, it only presents the fragment of proteins, right, which are processed by proteosomes after degeneration, right? So let's suppose it is presenting the fragment of core protein or capsid protein of hepatitis B virus. This is just a way to show the immune system that what is going on inside these cellular protein synthesizing factories, right? It is like a sampling system that they have to show a sample of their proteins to immune system that, hey, this is the protein that we are synthesizing, this is not something dangerous that is going on, right? So this poor cell, because its factory has been hijacked by virus and it has started producing viral proteins, so factories has been hijacked. So what will happen? So because normal protein is not being synthesized, immune system will catch it, right? Actually, cytotoxic T cell or CD8 positive T cells, CD8 positive lymphocyte, they are wandering in blood. Millions of different cytotoxic T cells with millions of different specificities, they are wandering in blood hungrily in order to find their prey, right? And somehow one of the cytotoxic T cell that is specific for this protein fragment that may come across in this infected hepatocyte, and it will recognize it and bind with it, right? And what it will do? It will do something horrible, but it has to do this. It will kill this cell. It cannot go inside the cell and catch this virus and destroy the virus. So it has to kill the whole cell. It will release some molecular grenades or granzymes, and with that it will kill the infected cell. So these poor infected cells, they have sacrificed themselves in order to protect their neighbors, the healthy hepatocytes. So the story does not end here, right? Just killing these factories, just destroying these factories is not the end of problem. There are still these virus particles strolling freely and happily. These virus particles that have already been created, they are still there, and they can infect a healthy hepatocyte. And then what will happen? The cytotoxic T cell will kill this infected hepatocyte, it will also generate more virus particle, then it will kill another hepatocyte, and in this way it may even end up killing the whole liver. And that's how fulminant liver failure occurs. When most of the hepatocytes of liver they are lost, this leads to fulminant liver failure. Of course we don't want that. We want the draconian measures to contain this virus. So what should be done? Not only cytotoxic T cell, or we call this cell mediated immune response. Not only this cell mediated immune response is required, we need something else as well. We need antibodies. We need antibodies to catch this virus and to kill this virus. Antibodies against what component of this virus? Against this capsid? No, against this antibodies against this capsid are not protective. Why? Because such antibodies, anti-HBC antibodies, they must penetrate through the envelope in order to reach this capsid and to kill this capsid, right? Which is not, of course, possible. So we need antibodies against these surface proteins. Antibodies against these surface proteins are protective. When they are attached, they will prevent the entry of virus, right? Also, these antibody tagged viruses, they will be quickly cleared by immune system. Macrophages will quickly come and destroy these virus particles. Naked virus particles, they won't be quickly destroyed. They will also be destroyed, but very slowly. But these antibody tagged cells, they will be quickly destroyed. So what we have learned up till now? We need cytotoxic T cell response, right, or cell mediated immune response, and we need humoral immune response through anti-HBs antibodies, right? The extent of damage and symptomatology of disease depends upon how efficiently our immune response behaves, right? And if immune response acts quickly, very few cells will be lost. Very few hepatocytes will be lost, right? And in such case, symptoms will be very less. It may be even asymptomatic hepatitis B infection. Patient may not even feel any symptom. Or if immune response is slow, many hepatocytes will be destroyed before the virus is cleared. And that will lead to much severe symptomatology, much severe hepatitis B infection, right? According to Greek mythology, there was a mythological figure with the name of Prometheus. Prometheus stole the secret of fire, and Zeus punished him by sending an eagle. This eagle ate the Prometheus' liver daily. A great part of Prometheus' liver was eaten by this eagle, but his liver regenerated every night. Every night this eagle ate the liver, and every night his liver regenerated. Probably the Greeks were also aware of the remarkable regeneration capability of liver. That if most of the part of liver is lost, it can then again regenerate very quickly and efficiently. So that's what the concept I want to tell you, that I want to impart in your mind, is that the main priority of immune system is not to save the liver, but to get rid of hepatitis B virus. The lost part of liver can then be regenerated, right? Unless the fulminant liver failure occurs. In that case, we need transplant, otherwise death may occur, right? It has been observed that virus does not cause a significant direct cytopathic effect. Hepatitis or inflammation of hepatocyte, it is caused when immune cell attack the virus infected cell, right? So my question is, why don't we just give immunosuppressive drugs to tackle this problem? Yeah, so in that case, virus will keep on multiplying. Actually, liver has lots of endoplasmic reticulum, lots of mitochondria. Even if its protein synthesizing capability is hijacked, protein synthesizing factory is hijacked, it can still continue somehow working because it has lots of organelles. But if lots of virus end up attacking the hepatocytes, hepatocyte will not be able to perform normal function. Furthermore, the small and but significant chronic inflammation will lead to cirrhosis of liver. Also, in the long run, it may cause hepatocellular carcinoma. So, we cannot allow the virus to continue replicating inside our beloved hard-working liver. So we must get rid of the virus, right? Another thing about hepatitis B virus is that if an HIV infected, if AIDS infected person, he gets hepatitis B or even hepatitis C infection, we must first treat the hepatitis before treating HIV. Why? Because if we treat HIV first, that immunocompromised person will become immunocompetent, and this immune cell, when they will see that their liver is infected, they will end up killing a lots and lots of hepatocyte, leading to fulminant liver failure. So that's why in such case, first we have to take care of hepatitis. First we have to reduce the viral load of hepatitis B or C virus, and then we have to treat HIV, right? So this was a relevant clinical point that is noteworthy here. So clearing the virus is not that simple and easy for body. This virus is very clever. It befools the both arms of immune system, the cell mediated as well as humoral immune response. So let's discuss first that how it tricks the humoral immune response, because it is easier to explain. You know, this virus creates subviral particles, and subviral particles are 1000 to 10,000 times more in number as compared to actual infectious particles, actual virus. These are fake viruses that are being created. These are only empty shells of virus. Now when body will create anti-HBs antibody, they will end up binding with these subviral particles, right? And that would be a useless activity. Meanwhile, it is binding with these subviral particles. This actual virus, it will slip away from the immune system. It will evade the binding of these antibodies. These antibodies are more likely to bind with this subviral particle than this actual virus, because these are 1000 to 10,000 times more subviral particles present, and this is called immune decoy. So these subviral particles, they tricks the immune system, right? Just like sometime we are trapped among fake friends or fake enemies, as in this case, right? So in this way, virus tricks the humoral immune response. It also takes care of the cell mediated immune response. Let's see how, but that is a bit tricky. Please listen it very carefully. So to understand this, we must first learn that how immune system works. We will briefly describe. So this virus, when it is created, there are macrophages, right? These macrophages have special receptors with which they can recognize, due to certain pattern, they can recognize the virus particles. Although they are macrophages, they are part of innate immune system, meaning that they do not have immunological memory. They still can detect the virus particles with a great accuracy with these special awesome receptors, right? What are these receptors? In Drosophila, or what they call it, fruit fly, right? In fruit fly, they found these receptors. They are also involved in embryogenic growth, and of course they have role in immune system as well. The scientists studying these receptors, probably they were so much fascinated by the working of these receptors, by the brilliance of these receptors, that they named them as great receptors or tall receptors, which is a German word which means great. When they found such receptors that were similar to these tall receptors in humans, scientists named them tall-like receptors. Why I am stressing on tall-like receptors, I will tell you later. So the thing is that with these tall-like receptors, these macrophages, they will bind with these virus particles, and they will engulf them. They will process their protein fragments and present their processed protein fragments on MHC type 2 receptor, not type 1, type 2 receptor. Type 1 receptor are present, type 1 receptors are on normal cell, while MHC type 2 or class 2 receptor are on professional antigen presenting cells like macrophages, right? So as they present them on, let's suppose this is again the fragment of capsid protein or core protein of virus. So of course helper T lymphocyte, again with millions of different specificities, are wandering in plasma, and one of the helper T lymphocyte that is specific for this protein fragment, this capsid or core protein fragment, it will come across this and it will bind with this MHC2 that is presented on macrophage. MHC2 presented antigen fragment or epitope, right? And it will become active. This helper T lymphocyte, or it is also called CD4 T cell. Why it is called CD4? Because it has a receptor that is called CD4. Simple as that. I'm not gonna go into details, then we will go into depth of immune system, which I don't want, right? So helper T lymphocyte helper T lymphocyte can differentiate into Th1 or type 1 helper T lymphocyte or Th2 helper T lymphocyte. Both are subset of or type of helper T lymphocyte. Th1 helper T lymphocyte, it promotes or it potentiates cell mediated immune response. It potentiates cell mediated immune response, right? And Th2, it potentiates and it promotes humoral or antibody immune response, right? Meanwhile, what was happening here? on these MHC1 bound antigen bound cells, what will happen? Specific cytotoxic T cell will come, but it cannot kill this cell unless it receives the order from this master orchestrator, this helper T lymphocyte. Helper T lymphocyte is commander-in-chief of immune system. Cytotoxic T cell will send chemical message in the form of interleukin 10. Interleukin 10. It will send the chemical message to this commander that, hey boss, I have caught something. Please order me to kill this cell. Please order me to kill this harmful cell. So this will then order back to kill this cell, and it will also order to make more and more copies of this cytotoxic T cell. And because it was presenting the cell and it was sending the message, more and more Th1 cell will be generated and Th2 will be suppressed. So less Th2 and more Th1, which will lead to more cell-mediated immune response. But as I told you, this virus is very clever. It knows how to tackle this problem. It knows how to handle this army business, right? It also generated e-antigens. And what e-antigen will do? E-antigen will be taken up by B cells, right? Actually, the process is not that simple. It is too complex to explain here. I am trying to simplify it as much as possible. So B cell, it caught this e-antigen and it will present it on its own MHC type 2. Again, it is also acting as professional antigen presenting cell. Now what will happen? Now here, let me remind you something. You know the gene of, what is this? Core protein. You know? Core protein. And before the gene of core protein, there was a small portion that was pre-core region, right? Pre-core region. And to synthesize e-antigen, the sequence started from here, from pre-core, and ended up to here, right? It means this e-antigen, it has most of the nucleotide sequence from here up to here, which is same as core protein, right? So it means if fragments of this e-antigen are formed, most of these fragments, they resembles this fragment, the fragment of core protein. And because of this reason, this B cell, it will attach and recognize and attach Th2 that were specific for capsid protein, core protein fragment, not another Th2, but this Th2, right? So what it will do? Now, because it has bound with this, it will generate more and more Th2 cell, which will lead to more and more humoral immune response, and it will suppress through inhibitory, I'm not gonna go into detail, through inhibitory chemical messengers or interleukin, it will inhibit Th1, right? So what is the point here? That e-antigen is trying to pull the immune response. Okay, it's like a tug of war. B cell is trying to pull, this is helper cell, right? B cell is trying to pull the helper cell towards what? Th2 side, to create more and more Th2, while cytotoxic T cell it is trying to pull it towards more and more Th1 or cell mediated immune response. Are you getting the point? But what will happen? What will happen? Who will win? Who will win? Th2 will win. Why? Because e-antigens are much more, they are much greater in quantity, they are much greater in number as compared to these MHC1 bound cells. MHC1 presenting these things, right? So because e-antigen are more Th2 will be produced, they will suppress synthesis of Th1 and they will cause generation of lots of antibodies against e-antigen or anti-HBE antibodies, right? And they will suppress the cell mediated immune response. Are you getting the point? So what will happen? Virus clearance will be slowed down. So in such circumstances, in order to clear the virus, first immune system will have to create enough antibodies to bind all these e-antigen and to clear all these e-antigen. And after all these e-antigens are taken care of, then it can generate Th1 cells and then it can create, what is this? Cytotoxic immune response. Otherwise, cytotoxic immune response won't be created significantly. And because this response is delayed, clearance of virus is delayed, and now more and more virus particles are created. Now it is much more difficult to get rid of virus. This virus is even more clever. What it will do? It can also, through e-antigen, it is some studies they also found that e-antigen, they can directly down regulate these great receptors, these toll-like receptors. So through these mechanisms, it has also taken care of cell-mediated immune response. And that's how virus evades the immune response. Now sometimes when e-antigen is found in blood, it means that virus is actively replicating inside the cell. Virus is freely living and enjoying the luxury of living and procreating inside the cells, right? So in such person in which virus is actively replicating, he or she is more likely to transmit such virus into another person. But sometimes virus very slowly replicates inside the cell, that very few e-antigens are produced and they are bound to antibodies and cleared, and they are not detectable in lab. It means virus is slowly replicating and transmissibility is less. Possibility of transmission to another person is less. So that's why e-antigen is also called transmissibility factor, right? And it is a good prognosis. If e-antigen is not found, it is a sign of good prognosis. But remember, e-antigen is not required for pathogenesis and replication of virus. They found some mutant strain of hepatitis B virus which do not produce e-antigen, but they are still infective. They are still pathogenic, right? So let's review what we have read. So for getting rid of this virus, we need cytotoxic T cell, which will kill the infected cell. And we also need this surface protein, antibody against surface protein, that will prevent the infection of healthy cell. This was the immune response, right? Now how it evades the immune response? By creating subviral particles which act as immune decoy, right? And also by producing e-antigen that does what? That produces B cell mediated more and more production of Th2 cell lines and decrease production of Th1 cell lines and decrease cell mediated immune response. Through subviral particles, it decreases humoral response. Through e-antigen and B cell, it inhibits, decreases cell mediated immune response. And it can also directly inhibit, what is this? Toll-like receptors. Thank you so much for watching this lecture. Please like this video and please subscribe to my channel and please give your precious comments regarding this lecture. Also, please check out my Facebook group. I will share the notes of these visual lecture series after the end of completion of the series. Thank you.